Hi everyone, welcome and thank you for joining us for today's seminar regarding microRNA biomarker discovery with respect to technologies, workflows, and data analysis. My name is Jonathan Shaffer and I'm Senior Scientist in Product Development at QIAGEN. Before we continue, I will give our standard disclaimer that the products described in this seminar are intended for molecular biology applications, and these products are not intended for the diagnosis, prevention, or treatment of a disease. For today's agenda, I want to cover the following information. We'll begin with a background regarding biomarkers, microRNA, and sample selection. We'll spend the bulk of the time today discussing microRNA isolation technologies, quantification technologies, as well as a circulating microRNA biomarker discovery experiment. We'll then discuss data analysis recommendations and finish up with a summary of QIAGEN's product offerings and field any questions that you may have at that time. So feel free to write in any questions that you have to the Q&A box, and at the end I'll read through these and we'll answer these live. So let's begin and start with the question, what is a biomarker? A biomarker is a characteristic that is objectively measured and evaluated as an indicator of normal biologic processes, pathogenic processes, or pharmacologic responses to a therapeutic intervention. In essence, it's a proxy for any of these. And a biomarker can be a characteristic such as a presence of a circulating antibody, abnormal blood pressure, blood cell counts, or electrolytes. Also, it could be a distinct histological indicator through immunocytochemistry or microscopy, abnormal liver function markers. It could also be the presence of a muscle injury protein, say in response to a heart attack, elevated kidney markers, or the gene status or gene expression status. Gene status, you could have a somatic mutation in a tumor, or for a gene expression status, you could have overexpression of an oncogene. Now, the method for analyzing your biomarker is linked to the specific biomarker. And for the presence of an antibody, you can use an ELISA. Obviously, for blood pressure, you can BP, you can choose a blood pressure cuff. Microscopy for histological indicators, various biofluid assays. And for the gene expression status, real-time PCR, next-generation sequencing, microarray, etc. And for somatic mutation, qPCR, or if you're in a discovery mode, next-generation sequencing. And so a lot of what we're going to discuss today is revolving around cancer biomarkers. And cancer biomarkers can be several different types. First off, a prognostic indicator, answering the question, is this likely to develop cancer? Also, a cancer biomarker can be of diagnostic nature, answering the question, what type of cancer is it? Also, and in the realm of collectively personalized medicine, a cancer biomarker can be predictive, it can be a pharmacodynamic, or it can be a recurrence biomarker, answering the questions, is this the optimal drug for my cancer? What's the optimal dose for my body? And will this cancer return? And I lump these in personalized medicine simply because in response to both the tumor, in addition to responding to the tumor type and the drug, this response will also be dependent on the individual's genetic makeup. Let's briefly discuss a non-invasive biomarker. And in order to use a biomarker for diagnostics, the sample material must be as easy to obtain as possible. And from the realm of easy to difficult, you have a urine or saliva sample, a drop of blood like those a diabetic patient would extract, a blood sample taken by a doctor, and more invasive would be a CSF sample, and most invasive would be obviously a surgical biopsy. So all things being equal, if you're looking at a non-invasive versus an invasive biomarker, you would like to go the route of a non-invasive. But again, everything has its place, including the non-invasive and the invasive biomarkers. Over the past few years, a lot has revolved around blood samples. And blood samples are made up of a cellular and acellular component, and nucleic acids, extracellular matrix components, and lipids can be extracted. And in addition, you can simply look at the blood cells for the presence of abnormal cells, abnormal cell counts, and also for stem cells or circulating tumor cells, for example. And collectively, Any of these components of blood can be biomarkers in and of themselves, or you can combine assays assessing multiple components to derive a biomarker panel. Now let's shift gears from biomarker and discuss microRNA. MicroRNA are approximately 21 nucleotide small non-coding RNAs, and here we have the canonical pathway of microRNA biogenesis. MicroRNAs are first transcribed by RNA pol II as a long primary transcript, which may contain more than one microRNA. In the nucleus, the prime microRNAs are processed to hairpin-like precursor microRNAs by the RNase 3-like enzyme TRACHA. The pre-microRNAs are then exported to the cytosol by exportin 5, where they're further processed by Dicer to mature microRNAs. The mature microRNAs are unwound, and one is incorporated into RISC, and microRNAs with high homology to their target mRNA can lead to mRNA cleavage, and microRNAs with imperfect base pairing to their target mRNA can lead to translational repression and/or mRNA degradation, and this is the case usually observed in animal systems. Let's briefly highlight some of the key findings of microRNA over the past 20 years. And it's nice to see the progression in a very small, condensed time frame. You have the initial discovery of microRNAs, particularly the first characterized microRNA by Victor Ambrose and colleagues, Lin 4. RNAi being characterized by Fire and Mello and C. elegans in 1998 that led to their 2006 Nobel Prize. After discovery, you then have characterization of siRNA as a tool, including large numbers of microRNAs being discovered. And at this point, you're looking at there were probably hundreds of microRNAs. Now, as of miR-based version 21, discovery of microRNAs continues to date, as there are more than 2,500 annotated microRNAs in human alone, and more than 35,000 annotated for all species included in miR-base. Then you have the first disease implications associated with microRNA and cancer by the Carlo Curci lab. You have microRNAs being used as diagnostics, and one of the key findings was microRNA as a serum biomarker in 2008. You have deciphering of the mechanisms of how microRNAs act, as well as the therapeutic implications of microRNAs as well. So you can see from discovery through siRNA as a tool to the first disease implications, diagnostics, mechanisms, and therapy, in 20 years or so, the microRNA exhibits itself as a natural phenomenon, discovery tool, and potential biomarker. Why should you quantify microRNAs? So if you exclusively look at the number of annotated microRNAs in miR-base, this reflects the interest in the field. You have the initial entries as of miR-base version 1, and as of miR-base version 21, I'd mentioned already that as of June 2014, there are more than 35,000 annotated mature microRNAs. Now, this number alone doesn't simply mean that there's interest in the field, that microRNAs have a very, very broad, sweeping role in biology, and changes in microRNA can be correlated with gene expression changes in development, differentiation, signal transduction, infection, aging, and disease. And looking at how expression analysis weaves into this, the microRNA expression analysis is the foundation for these discoveries. If you look at the role of microRNA in cancer diagnosis, microRNAs are being used to differentiate cancer tissue from normal tissue, to identify tissues of origin for cancers of unknown primaries, to distinguish tumor subtypes, to predict cancer predisposition, and also as minimally invasive biomarkers for the early detection of cancers. And this is a very hot area at this time. If you look at circulating microRNA biomarkers in the press, there is certainly no shortage of candidates. This is simply a truncated list from one publication, but we have microRNAs being described to be decreased in gastric cancer, decreased in non-small cell lung carcinoma, increased in breast cancer, increased in colorectal cancer, so on and so forth. And these also possess both diagnostic and prognostic value. So now let's discuss the type of samples that you will use in microRNA biomarker discovery. These can be your serum plasma, cerebrospinal fluid for circulating biomarkers, and you can also use your standard cell FFPE samples, urine, and potentially any fluid that relates to your particular disease of interest. And your analytes can be your cell-free microRNA, mRNA, or DNA. And for today's talk, we will be focusing on microRNA. So here's a question. What is blood? Blood is your red blood cells, white blood cells, platelets, and other cells such as circulating tumor cells. Serum is your blood, a separation of your liquid phase post-clotting, and plasma is separation of your liquid phase without clotting. Now, in plasma, high levels of nucleases are present, and as a result of this, you would say that freely circulating nucleic acids should be rapidly degraded. However, somewhat maybe surprisingly, stable nucleic acids can be detected in serum and plasma, meaning that they're protected by one mechanism or another. And how are microRNAs stable in circulation? Over time, it's an evolving story. So microRNAs can be protected in exosomes and other microvesicles, complexed with AGO2, protected by HDL, or complexed with another quote-unquote protective protein. In addition, I've included some references here at the bottom for your further reading. Let's take a closer look at exosomes. With exosomes and microvesicles, it's truly the cargo that matters. Exosomes are 50 to 200 nanometer small vesicles excreted by all cells, and exosomes are found in all biofluids, for example, blood. And exosomes contain stable RNAs such as mRNAs, microRNAs, and other non-coding RNAs, as well as DNA and protein, and this is all protected from degradation by a lipid bilayer. And the contents of an exosome are specifically packaged and are used as a mechanism of both local and distant cellular communication. There are many potential uses for exosomes as an analyte that hold promise for both disease detection and monitoring. Exosomes are involved with tumor profiling. They can be surrogate diagnostic markers for biopsy profiling and also hold possible utility to screen populations. Elevated levels of exosomes have been determined in blood of patients with late-stage ovarian cancer and lung cancer, holding promise for prognosis and therapeutic monitoring. Exosomes can be used to determine molecular signatures indicative of responses versus non-responses to therapy when you're profiling patients. Exosomes have therapeutic implications simply because you can deliver an exosome payload to specific cells. And lastly, exosomes are less of a needle in a haystack. There are 10 to the 12 exosomes per ml of plasma, while there are only 1 to 10 circulating tumor cells per ml of blood, and this has impact for potential clinical utility. So looking a little bit closer at exosomes and the role of liquid biopsies, let's take a microRNA example. So there are various publications, and this is certainly not an exhaustive list, where the exosomal microRNA expression parallels originating tumor cell expression. And this is minimally invasive simply because you can use exosome microRNA profiling in the absence of tissue to accurately reflect the tumor's profile. And the take-home message is that exosomal load assessment and exosomal molecular profiling hold great promise for both disease detection and monitoring. Now that I've spent a few slides on exosomes, let's ask this question. Should you only look at exosomes? And the answer is no, well, sometimes. And I say it that way because potentially 90% of microRNAs in circulation are present in a non-membrane bound form consistent with a ribonucleoprotein complex, meaning they're outside of exosomes. At the same time, though, analyzing the exosome fraction could maximize your signal-to-noise ratio of a potential biomarker, providing better sensitivity. Now let's move into the variety of technology. So I want to introduce some technologies that you'll use, and we'll spend maybe 15-20 slides or so on this before we move into a biomarker discovery experiment. So I'll explain the tools, and then I'll show you how to use the tools. So first off, for total RNA isolation from body fluids, you would use the microRNAs serum plasma kit. This is for purification of circulating RNA from plasma, serum, cerebrospinal fluid, urine, saliva, etc. Virtually all biofluids can be used in conjunction with the miRNA serum plasma kit. Some of the highlights of the kit are that it includes a synthetic RNA control for normalization. It provides you with a minimal elution volume, and the RNA is highly pure for all downstream applications. As you can guess, it's easy and robust and can also be automated. And our one recommendation when working with serum and plasma is, when possible, avoid heparinized plasma simply because heparin is a potent reverse transcription inhibitor. However, if you can't avoid heparinized sample, just let me know. At QIAGEN, we're always exploring new avenues to access new samples, so we certainly have a solution for you if you have heparinized plasma. So at the end, contact me, jonathan.shaffer@qiagen.com, and we can set up a solution for you. Now back to task here. With the miRNAs serum plasma kit, we have the workflow on the left. You add your Chiazol lysis reagent, add chloroform and separate your phases, and you add ethanol to the aqueous phase and bind this to a column, wash it, and elute your ultra-pure RNA. The next kit I would like to discuss is for exosome purification and total RNA isolation from serum and plasma. This is the ExoRNAs Serum Plasma Maxi Kit. This specifically enriches for RNA contained in vesicles. You can quickly isolate purified total RNA from microvesicles, and you can efficiently isolate both mRNA and microRNA from plasma and serum. And this kit also enables use of high input volumes for sensitive detection of low abundant transcripts. So what is the input range? The input range is 200 microliters to 4 mLs of serum or plasma. So if you look at how ExoRNAs prep compares to an ultracentrifugation prep, we have the following example. So if you use a scanning electron microscopy to 20,000x magnification, this reveals higher purity with the ExoEasy kit. Both preparations contain your vesicle-shaped structures within an expected size range. However, ultracentrifugation, you also have many smaller unidentified structures that do not match the expected size. And the ExoEasy column, which is part of the ExoRNAs kit, you have intact vesicles with higher purity. Here we have the workflow of the ExoRNAs kit. Can be divided into 3 phases. Phase 1 is where you separate your serum or plasma. Phase 2 is then where you isolate your exosomes. And phase 3 is where you isolate your RNA over a column, very similar to the miRNAs serum plasma kit. Now, the greatest advantage of this, in addition to higher purity, is also the time. So that microvesicle isolation can occur in 20 minutes, and RNA isolation takes a little more than half an hour. So collectively, from sample to isolated RNA, you can have isolated RNA from exosomes in less than 1 hour. So I told one of the organizers here, let me take my advertisement here for Coke. So let's continue on. So now let's discuss circulating microRNA biomarker discovery. And with this, you will use the microscript PCR system. This is a real-time PCR-based approach system, and the workflow is as follows. You isolate RNA, perform reverse transcription, prepare your PCR premix, load your PCR arrays or individual primer assays, perform real-time PCR, and analyze your data. And let's go into more detail of the components of the microscript PCR system. So for reverse transcription, you will use the microscript 2RT kit, and the kit is built with your particular biological questions in mind. If you're interested in biogenesis studies, perhaps which precursor microRNA your mature microRNA is derived from, and also what mRNA your microRNA is potentially interacting with, you would want to synthesize cDNA using high flex buffer, which allows you to detect all RNA molecules from one cDNA synthesis. Now, for mature microRNA quantification, profiling, and biomarker discovery, we've developed the specific solution for this type of studies. You would synthesize cDNA using high-spec buffer. High-spec buffer is a patent-pending technology for the specific detection of mature microRNAs. The reverse transcription of longer RNAs that sometimes give you a background cross-reactivity signal when profiling mature microRNAs is highly suppressed, leaving you with a very ultra-clean, mature microRNA-specific signal. Now looking more at the second component of the microscript PCR system, this is the microscript microRNA PCR arrays. Basically, these are individual mature microRNA primer assays that are pre-dried in PCR plates. These are pre-formatted single-use plates with wet lab verified assays. And just as an overview, we offer arrays for micronome profiling, targeted micronome profiling, or bioinformatic-driven profiling. And I'll go over these in more detail in the coming slides. But the greatest advantage of these are that you simply prep your PCR reaction mix, load your plate, and run your real-time experiment. There's no individual pipetting of primers. So I mentioned that the assays are wet lab verified. So what does assay validation mean to you? It means a total commitment to quality. So our workflow is as follows. The primers are designed using a state-of-the-art proprietary algorithm. And following this, all primer designs are individually bench validated in product development to ensure negligible background signal, single specific PCR products, optimal dynamic range, and optimal reaction efficiency. Basically, everything that you come to expect out of a primer, we test it. Any primer that then fails validation is redesigned and retested. And if the primer fails again, we don't offer it for sale. Also, just as a note, all lots of microscript PCR arrays are individually quality controlled. So let's discuss the variety of options you have now. We have the miR-based profiler microscript microRNA PCR array. And what this enables is you to choose your micronome. We have coverage for human and mouse through miR-based version 21, with over 2,400 assays for human alone, over 1,700 for mouse, and we also have coverage for rat, dog, rhesus macaque, cow, and our latest species of interest is horse. Some of the benefits are, again, that the assays are 100% validated. And the best advantage to you is that the solution is completely scalable. You can choose as many plates as you want of what your question is that you want to answer and also how it fits in with your budget. So if you want to be on the cutting edge of discovery, you can profile the version 21 micronome. However, if you want to profile only the tried and true original microRNAs, you can choose to only profile the version 16 micronome and cut down your number of plates. So it's truly scalable based off of your type of experiment that you want to do. Now you may say, are the most recently annotated mature microRNAs even expressed? And we did this experiment where we profiled a liver tissue sample which is a pool of 10 healthy male liver tissues across our 2,400 assays. And as you can see from assay 1 through 2,400, we have the raw CT values included. And as you can guess, that even the most recently annotated mature microRNAs are also exhibiting signal, meaning that they may be relevant. So what we do is we offer as many assays as we can possibly validate and let you decide how it fits in with your experiment and whether they are relevant for your experiment of interest. And this is just a comparison of coverage that we have in comparison to other leading vendors, that we have coverage through miR-based version 21 for microscript, while the other two vendors that I have listed have coverage through approximately miR-based version 10. Looking at this in one more manner, the same liver tissue profiling experiment. For each plate, the expressed microRNAs were determined, and this is a CT with a limit of detection of 35. And as you can see, you have approximately 300 in plate 1 and around 200 for plates 2 through 5 and 300 plus for plate 6. Now this is microRNAs included in miR-based version 19, version 20, and 21. And as you can guess, that's quite a lot that are actually expressed. Also note, miR-based, plate 7 is actually a partially filled plate. And on the right, we have the plate median and plate mean for each of the particular plates. And beyond plate 1, which, giving their due, they're the original tried and true microRNAs, plates 2 through 7 all have approximately the same mean and median. So when you move beyond the original microRNAs, take great notice of these because they may be waiting to be discovered for the next great biomarker discovery. Let's take a look at our targeted micronome profiling. Our targeted micronome profiling blends bioinformatics with our leading content. The MI-Finder array collects the 372 best expressed and best characterized microRNAs into one array. The serum and plasma blends in-house profiling experiments we've done with our bank of content assays, where we include the 372 best expressed microRNAs in serum and plasma. The Cancer Pathway Finder collects the 372 microRNAs most broadly associated with cancer into one array, and the Liver MI Finder is another one where we've blended in-house wet bench experiments with our content, and these profile the 372 best expressed microRNAs in liver tissue. And the last type of content is our bioinformatic-driven profiling. We have teams of scientists at QIAGEN that use state-of-the-art bioinformatics and manually curate literature to organize microRNAs into pre-formatted panels based off of the hottest areas of biology. And we have the MI Finder that collects the 84 best expressed and best characterized microRNAs into one array, a variety of cancer-focused arrays, a hypoxia array, neurological development and disease, tumor suppressor microRNAs, and also a serum and plasma array. Now, this serum and plasma array collects the 84 microRNAs most widely published to be both present in circulation and differentially expressed in response to disease. Now, you may say, well, that's all well and good that you have identified these microRNAs that are the hottest areas. However, this is nothing that I'm interested in. And the good news is that these arrays are fully customizable. We can place you in contact with a content development scientist and have an array developed for your specific needs. So let's take a look at the formats. In addition to our 96 well, where this contains 84 microRNAs plus 12 controls, we have a 384 well 4x96 format. And what this is essentially 4 copies of a 96 well in a 384 well plate. So if you have access to a 384 well instrument and a 96 well instrument, please use the 384 well instrument, simply because you can either increase your workload fourfold, or you can simply do your work all at one time and go home early in the day. And I like to tell people that it really depends on the day of the week. So Monday through Thursday, you can increase your workload fourfold, and on Friday, go home early. In addition to these two 96 and 384 well format, we also have our 384 well HC format that allows you to assess 372 microRNAs plus 12 controls. In addition, we have pre-formatted content for QIAGEN's Rotor GeneQ. Due to our variety of plastics and formats, we have compatibility with a wide range of instruments from Life Technologies, Bio-Rad, Eppendorf, Roche, Agilent Strategy, Takara, as well as QIAGEN. And a couple more products I'd like to introduce you to. For limiting samples, we have the microscript preamp kit. And for a micronome profile, you can use as little as 1 nanogram of total RNA. This is a highly multiplex PCR-based preamp and is compatible with all microscript arrays and individual primer assays. And I'd already mentioned cells or tissues, 1 nanogram of total RNA is the limiting amount. And for fluids, serum or plasma, you can use this with 50 microliters or less. And when you're working with urine, cerebrospinal fluid, or aqueous humor, any amount we recommend preamp. And using microscript preamp in a quote-unquote verb sense, when in doubt, microscript preamp. We have customers all over the world that are using preamp by default. And if you're unsure, go ahead and preamp it. Now, the last product I'd like to introduce you to is a high-throughput microRNA expression profiling platform, the microscript microfluidics. And what we've done is that we've developed a whole host of reagents that are highly suited and highly optimized to enable microRNA profiling on the fluidine biomarker. And collectively, this represents the first complete system for microRNA expression profiling on the biomarker. Now, the take-home message is that what's the bottom line advantage? It's a savings of both time and money. If you're unfamiliar with the biomarker, you can interrogate 96 samples at one time, each with 96 assays, leaving you with 9,216 data points in one real-time PCR run. So let's put this to the test and see the savings. On the left, if you have 96 samples and 384 assays and access to a 384 well instrument, you need to perform 96 real-time PCR runs. And at little more than 2.25 hours per 384 well plate, you can achieve your nearly 37,000 data points in 216 hours, roughly speaking 27 days. However, if you use a fluidine biomarker, you can achieve the same experiment with only 4 chips. And very generous here in time, very conservative in time, 5 hours per chip, you can achieve your 37,000 data points in 20 hours, which is a more manageable 2 days. Now let's quit talking about products and discuss some of the workflows that you'll use. Workflow 1 would be when you're working with PCR arrays or assays with non-limiting amounts of cells, tissue, or serum or plasma, which is approximately 100 to 200 microliters. Step 1, you convert your microRNA to cDNA. Step 2, you prepare your PCR premix and aliquot it across your array. Step 3, you run your real-time experiment. And Step 4, you analyze your data. Workflow 2 is where you incorporate preemp into your workflow. This would be again for limiting samples such as limiting cells, tissues, small amounts of serum or plasma, perhaps 50 microliters or less, or other fluids. Step 1, you convert your microRNA to cDNA. Step 2, you perform your multiplexed preemp. Step 3, you prepare your PCR premix and load your plate. Step 4, you run your experiment. Step 5, you analyze your data. Workflow 3 is specific for serum and plasma expression profiling. Step 1, you collect your whole blood and separate your serum or plasma. And tip 1 is, if possible, prep your sample, prep 100 to 200 microliters of your sample for isolation. And tip 2 is, though, if you don't have that type of input volume, you can use this kit to isolate RNA from as little as only 5 microliters of serum or plasma. In step 2, once you have your sample separated, isolate RNA using the miRNAs serum plasma kit. Step 3, you perform reverse transcription. And step 4, perform preemp if your experimental sample is of less than 50 microliters. And step 5, perform real-time PCR. And our array recommendations, in addition to individual primer assays, are the miR-based profiler micronome array, the serum and plasma 384HC targeted micronome, or the bioinformatics serum and plasma array. Step 4 is when you're working with urine, cerebrospinal fluid, or other fluid microRNA expression profiling. Step 1, you collect and clarify your sample, and our recommendation is still to start with 100 to 200 microliters, but this can be used with down to 5 microliters. Step 2, isolate your RNA using the miRNAs serum plasma kit, plus spike and control. And the tip here is that the spike and control enables calibration of your samples based off of RNA recovery. And we'll go over this in more detail a few slides from now. Step 3, perform reverse transcription. Step 4, perform preemp by default. And Step 5, perform real-time PCR. And our array recommendations, in addition to the miR-based profiler micronome array, are to use the miR-based profiler targeted micronome array or the bioinformatic miFinder array. Now, Step 5 is a concept. It's the biomarker discovery workflow. And we'll go through this, and then this is what we're going to use then to discuss our case for the day. So phase 1 is where you determine your expressed microRNAs. This is a qualitative yes or no assessment. And the best way to do this is to use a pooled sample profiling approach. We recommend incorporating up to 10 samples per pool and do pools of both normal and disease, anywhere from 2 to 4 pools per normal and per disease. And for this, you want to use maximal number of assays. This can either be micronome profiling with real-time PCR, or you can use next-generation sequencing. But in phase 1, we're simply wanting to know what is present. And basically what we want to do is separate the expressed from the non-expressed and narrow our field to maximize both our time and resources. Phase 2 is then where you use your microRNAs that were expressed in phase 1 and determine your differentially expressed microRNAs. And as a first set of samples, you can use any of the samples that went into your pooled samples to do individual profiling on. And for this, you're only screening your expressed microRNAs from phase 1. And in phase 2, you want to then determine your differentially expressed microRNAs as well as your invariant microRNAs, because the invariant microRNAs will be used in phase 3 for data normalization. So in phase 3, this is your classifier refinement. This would be further individual sample profiling only on your differentially expressed microRNAs from phase 2, as well as some invariant microRNAs. And I can't stress it enough to include invariant microRNAs, simply because there's nothing worse than having to go back and redo previous experiments because you weren't able to identify a normalizer. And our recommendation is to include anywhere from 4 to 6 invariant microRNAs, just to hedge your bets that you've actually selected an invariant microRNA that will remain invariant through your entire sample population. So let's now discuss our case example that we want to go over today. This was a liver toxicity microRNA biomarker project with Dr. James Deere from Edinburgh University, and it revolved around acetaminophen overdose. And acetaminophen overdose is a common poisoning worldwide and can cause acute liver failure, potentially resulting in acute liver failure and death. And the scope of the collaboration was to determine microRNA markers of acetaminophen poisoning and also to distinguish acetaminophen poisoning from other liver syndromes. And we followed the three-phase workflow. Phase 1 we completed where we identified 377 expressed microRNAs, and we narrowed the list down from over 1,800. We did this a couple years ago, so we were only using miR-based version 18 micronome. Phase 2, where we determined our differentially expressed microRNAs, we found 92 microRNAs of interest and narrowed this down to 16 microRNAs using a classifier algorithm. Phase 3 is ongoing, and we're refining the initial classifier, including using the assessment of blind samples. So let's go over this in more detail. So Phase 1, again, you're determining the expressed microRNAs and essentially surveying the microRNA landscape. For this, we started with 54 samples, 27 control samples, 27 liver injury samples. We isolated RNA with a microRNA serum plasma kit and prepared 4 pools, 10 samples per pool, 2 controls, 2 patient pools. And for expression profiling, we used the miR-based profiler micronome array, miR-based version 18 of 1,800 assays. And for this, we selected microRNAs that were present in all pools, 3 pools, 2 pools, and 1 pool, and arrived at a population of 377 assays. So we narrowed this down from 5 384 well plates to 1 384 well plates simply by performing a pooled approach. Phase 2, we determined our differentially expressed microRNAs. This was a secondary screen of the individual samples from Phase 1. Again, we used the 54 samples from Phase 1, but we profiled each one individually. And we used the microscript PCR system with 377 assays on the fluidine biomark. So we went through 54 samples, which roughly translates to 40,000 data points in 4 days. And once we got more familiar with the fluidine biomarker system, we've cut this down to 2 days. And what we've identified is microRNAs that were differentially expressed, including ones that have been published in association with acetaminophen poisoning, including miR-885-5P and miR-122-5P. And if you look at these on a volcano plot, where we plot fold change versus p-value, we're able to see microRNAs that were significantly upregulated. Phase 3 is the statistical power phase. And this would be where you're further refining your classifier algorithm. And you're screening a larger cohort of affected individuals, and you're rearraying the differentially expressed microRNAs and looking across additional samples. And you're continuing to refine your classifier model. And in the bottom here, this is the original 54 samples with the top 16 microRNAs. Now, some important considerations when performing a biomarker discovery development experiment. Our first recommendation is to be novel. Initial whole micronome screenings can leave you with unique signatures. And for this, let's take this a retrospectively what if question. So what if the group we worked with had only profiled the first 2 plates, roughly 768 mature microRNAs? Of the 92 differentially expressed, they would have only identified 64, and this would have translated to a 30% loss of data, and actually they would have lost 2 invariant microRNAs. And don't underestimate the importance of this. Invariant microRNAs are critical to the success of your experiment. Without these, you won't be able to properly normalize your data. Also, 5 microRNAs from the optimal 16 microRNA signature would not have been assayed, including 3 of the top 5. So moving on, some other important considerations are that you want to verify your microRNA signature on naive samples, and strong changes might be general indicators or even be non-specific. They could be associated with other liver disease or potentially even stress markers. Also, note relatively weak changes might add specificity, and as you could guess, high levels of false positives are undesirable. But you also, lastly, want to ask the question, what's next? And with that, you want to determine the function of your microRNA. You can revisit literature, Ingenuity IPA from QIAGEN, etc., and potentially screen other types of samples to help define specificity as well as refine your signature. So now let's discuss some data analysis when working with serum and plasma samples. Serum and plasma samples have a unique special case simply because the small nucleolar control of panels does not exhibit robust expression and should not be used as normalization controls. And small nucleolar RNAs are typically used for normalization when working with cell and tissue samples, but these don't fit the bill when working with serum and plasma. Typically, you see these type of CT values well into the 30s or even potentially not expressed. So you may say then, what are my options? So step one, we recommend calibrating your sample using the C. elegans miR39-3 PCT mean, and then normalizing using one of the following two methods. Option one, normalize your CT to the CT mean of all commonly expressed microRNAs, or option two, normalize to the CT mean of invariant microRNAs. So for this, step one is that you want to calibrate your data. And you calibrate your data using the C. elegans miR39-3P. These are found on every PCR array, and what this assay detects is the microRNA serum plasma spiking control that has been added during sample prep. So if you look at an example here, sample 2 versus sample 1, all assays in sample 2 appear to have a delayed CT value. But at the same time, C. elegans miR39-3P also has a delayed CT value. And your conclusion is that you should calibrate your samples because the C. elegans is indicating a differential recovery. So how should you calibrate this? You should calibrate this by taking sample 2 and subtracting 3 CT units from each assay in sample 2. Don't mix and match. Be sure that you do this with each microRNA and also indicate this when you're writing this up for publication that you've corrected for differential recovery due to sample prep. And once you correct this, you can see that there's virtually no CT difference for miR16, 192, or 25. And instead of no CT change for miR103, it was actually more robust in sample 2. And miR21, instead of a 4 CT delay, you're only looking at a 1 CT delay. So once you've calibrated your data, how should you normalize this? Option 1 is to normalize to the CT mean of expressed microRNAs. And this involves calculating the CT mean for commonly expressed microRNAs, and those are microRNAs with a CT value less than your limit of detection in all of your assessed samples. So to be included in this calculation, it needs to be expressed in all samples. Option 2 is to normalize to the CT mean of invariant microRNAs. And this involves calculating the CT mean for invariant microRNAs and choosing at least 4 to 6 for this calculation. Now we have a chart here, commonly expressed microRNAs. Don't take this to mean that these are invariantly expressed. Invariant expressed microRNAs that can be used for all types of serum and plasma samples is the holy grail. A lot of resources are being put into this worldwide, but there still is no definable housekeeping microRNA in circulation. These are simply microRNAs that you can first look at to see if they're invariantly expressed. But the best case is to always perform a broad micronome profile or with PCR or NGS to determine differentially and invariantly expressed microRNAs. When you compare option 1 to option 2, which should you choose when? So option 1, commonly expressed microRNAs, choose this when you're working with a micronome panel, a targeted micronome panel, or a pathway-focused panel. This would be your large panels of microRNAs where the plate mean is truly indicative of differences in RNA amount. Option 2 is the invariant microRNA panel, and you would choose this for small panel screening or individual assays. As a note, in this example, the fold regulation looks highly similar, irrespective of the method, because your results should be as independent of your chosen normalization method as possible. And as a second note, for small panel screening, do not use the CT mean of microRNAs, as this array is biased, meaning that your microRNA assays included on this array are not at random. So let's finish up with some resources. Here we have a summary of QIAGEN's microRNA portfolio. We have a whole host of isolation technologies for cells, tissues, FFPE tissues, serum plasma, exosome isolation and enrichment of RNA, and we also have PAX gene solutions for your blood types and blood and tissue types. For quantification and profiling, we offer the microscript PCR system, and for functional analysis, we offer a host of transfection reagents, microRNA mimics, and inhibitors. Now, for biomarker discovery, sometimes your vision exceeds the capacity of your lab, and I'd like to remind you about QIAGEN Service Corps on multiple continents that we have scientists highly adept in total RNA isolation, real-time PCR, and data analysis is included as well. I'd like to remind you of the QIAcube for automated sample prep, and the QIAgility for automated PCR setup, and the RotorGene-Q for real-time PCR. Where can you find the products I discussed today? QIAGEN.com or QIAGEN.com/GeneGlobe. At GeneGlobe, you'd simply type in your microRNA of interest, and you receive a whole host of products that are associated with it. And with that, let's take a look at the questions. I'd like to thank you for attending today's seminar. I truly appreciate it. I'd like to also encourage you to write down my email, jonathan.shaffer@qiagen.com. At any time, feel free to write me any questions that you have with respect to microRNA or with respect to products that you may need or even your workflows. So with that, let me take a look at the questions. First question we have, is there any pharmaceutical products in the market having microRNA? They are actively in development. There are molecular diagnostics being associated with microRNA. We have a second question here. So for biomarker discovery workflow, how many samples do you recommend per pool? And this truly depends on your number of initial samples, but our recommendation is up to 10 per pool, maximally 3 normal pools and 3 disease pools. Okay, we have another question here. Can you elute exosomes from the exo-easy membrane without using chiazole? Meaning that we want to isolate intact exosomes. And the answer is no, not at this time. But we are developing a solution for this. Okay, you mentioned data analysis solution. We have another question. And my question is, what is the cost of the data analysis? And the answer is that it's actually free, that it's freely available and can be accessed through the web. And you can access it at QIAGEN.com/GeneGlobe. Sure, we have a question. Is there literature resources available for the comparison and normalization that you mentioned? So yes, this is actually, I can provide you with this. Simply write down my email, jonathan.shaffer@qiagen.com, and I can point you to a particular paper from the group that was involved with the MICE guidelines. And these are methods of normalization that are recommended by this group. Okay, we have a question about heparinized plasma, and the question is that their samples are heparinized. What are their options? Okay, so what we have is we have a preliminary protocol where we basically remediate the heparin, and if you're interested in this, feel free to email me and I will forward you the protocol. Okay, another question we have is is it acceptable to normalize exclusively to C. elegans that has been spiked into a serum prep? Okay, I see what you're saying. So the recommendation is to calibrate using the C. elegans and then normalize to the plate mean or invariant microRNA. Normalization to C. elegans miR39 only accounts for differences due to sample prep and doesn't have any biology tied to it. One more question. Would you please remind me what your recommended starting volume is for serum? So our recommended starting volume is 200 microliters, but if you incorporate preamp into your workflow, you can isolate from down to 5 microliters. Okay, so that looks like that's it for today. Again, thank you everybody for attending, and feel free to write down my email, jonathan.shaffer@qiagen.com. Thanks everyone, have a great day.